Esteemed jury, dear participants, listeners, ladies and gentlemen, on behalf of the Students' Scientific Society of Medical University of Silesia in Katowice, we would like to thank you for coming and taking part in the International Medical Congress of Silesia 2024. My name is Mikołaj Górka, and it's my great pleasure to coordinate the experimental medicine session with my dear friend, Joanna Smolarczyk. We'd like to cordially welcome the esteemed jury of our session, President of the jury, Professor Agata Stanek, and members of the jury, Dr. Anna Pacwa and Dr. Aleksandra Boldys. Dear participants, before we start the first prelection, let me explain you the principles and rules of the session. The orders of the presentation will be the same as in the abstract book published before the conference. The maximum time from presentation is 7 minutes and 3 minutes for the discussion. If the presenter exceeds the time of the prelection, he or she needs to move on to the conclusion and finish the presentation as soon as possible. Don't be afraid, we will let you gently that the time has just passed. Please remember that all the presentation and discussion must be run in English.
Esteemed jury, I would like to kindly remind you that if you are the supervisor of the paper which is presented, you should not assess the work. In that case, please cross out the number on your card. Of course, other members of the same clinic can assess the presentation and the assessment will be verified. We kindly ask the jury to assess 4 aspects of each prelection. Scientific value from 1 to 6 point, way of presenting 1 to 6 point, discussion from 1 to 6 point, and on contribution of the participant. When the session ends, we kindly ask you to give the cards back to the coordinator. These are all rules and regulations that we need to observe. But if something is unclear, I would be glad to answer any question you might have. Sorry, not to 6, only to 5, from 1 to 5 points.
Yes, we have 3 minutes for the discussion, and we will pass you a microphone after the presentation.
Okay, so if everything is clear, I think we can start with the first presentation.
So at the beginning, we would like to ask Natalia Frankowska to present the paper entitled Characterization of Bacteriophages from Clinical Strains of Clostridioides difficile.
So, hello everyone, one more time. My name is Natalia Frankowska. I am a PhD student from Medical University of Gdańsk at the Department of Molecular Bacteriology. So today I would like to mention you briefly about the characterization of bacteriophages from clinical strains of Clostridioides difficile. So C. difficile is gram-positive, strictly anaerobic and spore-forming bacterium. And C. difficile infection can happen in a situation when gut microbiota is disturbed, usually after prolonged antibiotic treatment, and it may cause diarrhea and colitis. In order to track how the bacteria spreads all over the world, it is classified into ribotypes, and we can also distinguish so-called hypervirulent ribotypes, and they are said to sporulate with higher efficiency, they produce more toxins, and they're also more antibiotic resistant. Bacteriophages are viruses that can specifically infect bacteria, and all known C. difficile phages or bacteriophages are temperate. It means that they can integrate to the chromosome of the bacteria and be in a form of a prophage. So C. difficile phages are characterized within a bigger project that investigates the role of prophages in terms of virulence of its host. So basically, we first start from the collection of clinical strains of C. difficile, and we aim to get around 500 of them. And we get our samples from Gdańsk and also from our collaborators from Medical University of Warsaw and Silesia. We also would like to collect the clinical data of patients together with the strains. So the next step is to screen for these functional prophages. Functional, I mean that these phages can exist outside the host in the environment in a form of viron. And then chosen candidates we choose for the isolation and sequencing of the DNA. And then with the use of CRISPR technology, we will remove the phages from the host genomes. And the most important step is to analyze the virulence between the wild-type strains of C. difficile and the prophage-free strain. And this step is divided into two studies. So in vitro analysis, this will include for example, production of toxin, phosphorylation rate, and also in vivo studies will be performed on in vivo mouse CD infection model. So basically, to identify these prophages, we still have to start with anaerobic culture of C. difficile. And then the phages are induced with antibiotics such as mitomycin C or norfloxacin. And after this step, we end up with the processed sample that is filtrated supernatant that consists of leftovers of bacterial genomic DNA and also of virions. And the next step is to get rid of this genomic DNA of bacteria with the DNase 1. And then the DNA is released from the head of the phages by simply boiling the sample. And then we perform the detection of phage DNA by PCR reactions. To detect phages, we use three different sets of primers. So we use primers to amplify fragments of choline genes of myoviruses and cephaloviruses. And also we use the primers to amplify fragment gene of so-called TPI gene, that is housekeeping gene for C. difficile. So this is the electrophoresis gel before DNase 1 treatment. And here we can see that we have some residues of DNA of bacteria. And also we have some potential phages from Myoviridae family and also from Cephaloviridea family. And after this DNase 1 treatment, we got rid of bacterial DNA left over. And here we have 3 functional prophages. And in our project, we were able to collect so far around 400 of strains, and we were able to test around 1/3 of it. During our studies, we also observed that most of these phages belong to Myoviridae family. Also, we observed very interesting dependence that actually most of these hypervirulent ribotypes of C. difficile, they actually contain prophages. So these are the ribotypes depicted in red, the ones I mentioned to you briefly in the introduction. And here is the first phage that was isolated by us, and this is the temperate Cephaloviridea phage with long non-contractile tail. And in its genome, we have found a few interesting genes. For example, this is the gene that encodes for endolysin. Endolysin is a phage protein that can lyse bacterial cell wall in a specific manner. And also the second gene encodes for the protein that resembles C. difficile binary toxin. And we would like to characterize these proteins more deeply in the future. And also in other C. difficile phage genomes, we were able to find many transcriptional factors whose function weren't fully investigated. And also we observed some genes that may encode virulent rate proteins, and we believe that they may have some connection with affecting the phenotype of the host. Okay, so to sum up, what we were able to observe so far that most of these functional prophages were actually induced from these hypervalent strains of C. difficile, and genome of these prophages may be the source of genes that affect their host, but also we assume that they may affect even the virulence, increase that virulence, but this is something we would like to confirm by the end of our project. And here is the list of all our collaborators, and the project was funded with Opus grant, and I would like to thank you for your attention.
So now I would like to invite you all to ask a question and discuss the presented paper. And I remind you that the question can be asked both by society committee as well as by all participants. So feel free to ask any questions. So I will pass the microphone to the jury.
Okay. So I have the first question. Does your results, do your results can help in treatment in the future? Does it make any difference for
Okay, so
clinical practice?
So from our preliminary results, we were able to actually observe that these prophages were in this hypervalent strain. So maybe in diagnostics, it can help to estimate what could be the course of infection by C. difficile, and also we would like to go deeply inside the genomics of these phages, because maybe with the genetic engineering we can let's say make the phages like lytic phages, so then they could be used in phage therapy. But this is something that could be done in few years. This is not something to be done like now.
Congratulations, very interesting presentation. Could you tell us what was your contribution to this work?
Okay, maybe I will show by the slides. So basically, this is the part that will be performed by our collaborators, and also this part is shared with the collaborators. But this one, this is let's say part of my contribution. Of course, sequencing is performed by external company. Also, I performed all of these steps and also these ones. Oh, and the ribotyping is this is also the cooperation with other university.
It's the topic of your PhD thesis, or not? It is a topic of your PhD thesis.
It's connected mostly, yeah. So these results, they will be included in my PhD thesis, but I also investigate phage-derived proteins. So I would like to purify these proteins and see how they affect with the bacteria. So this is the part of it.
Thank you.
Thank you.
Thank you. Thank you for your presentation. And now I'd like to invite Alexandra Lorek from Wrocław University of Science and Technology with titled Method of Doping Selected Organic Matrices with 4 Chosen Photosensitizers.
Hello. Am I visible?
Oh, hi, you are blurred. So if you can.
Okay.
Okay, now it's okay. Thank you. Could you share with us your presentation?
Sorry, I have a note that I can't share the screen while it's shared right now.
Okay. Could you try now?
Yes, it's working. So good morning, ladies and gentlemen. My name is Alexandra Lorek, and I'm delighted to have an opportunity to present my research findings here today at International Medical Congress of Silesia. I'm from the Wrocław University of Science and Technology, where I'm affiliated with the Faculty of Fundamental Problems of Technology. Today, I will discuss an innovative method of doping selected organic matrices with 4 chosen photosensitizers, which holds potential for advancing photodynamic therapy. Analyzing the development of photodynamic therapy, we identified the potential of readily available and inexpensive organic matrices, and these matrices can help minimize complications post-therapy. Photodynamic therapy uses chemical photosensitizers and light irradiation at specific wavelengths to eradicate target tumor tissues. Although photosensitizers are non-toxic to cells in the absence of light, when irradiated with specific activated wavelengths, photosensitizers generate cytotoxic singlet oxygen that destroys cells. This research topic is particularly significant to me due to my internship at Kamińskiego Hospital in Wrocław, where I observed the challenges faced by oncology patients. Our aim was to evaluate agar, starch, and gelatin matrices as neutral carriers of photosensitizers in photodynamic therapy, with particular emphasis on optimizing therapeutic effectiveness while minimizing patients' discomfort and chemical exposure using spectroscopic analysis. We prepared organic matrices by doping them with photosensitizers and optimized the measurement system. Spectroscopic measurements were conducted to analyze absorption and luminescence spectra in the reflection system, providing insights into their optical properties for photodynamic therapy. And we selected agar, gelatin, and tapioca derived from cassava as our organic matrices due to their biocompatibility, affordability, and availability. And these materials are widely used in biomedical applications and offer unique properties that make them suitable carriers for photosensitizers. And for photosensitizers, we chose phenolphosphate A, chlorine E6, 5-aminolevulinic acid, and protoporphyrin 9, based on their proven effectiveness in photodynamic therapy and their varying chemical properties, which allowed us to study a range of interactions with the matrices. And chlorine E6, a naturally occurring photosensitizer obtained from alga, has found its primary application in PDT for the treatment of malignant tumors. It's commonly used especially for lung cancer, metastatic breast cancer, and refractory ovarian cancer. Formed using a 2 milliliter syringe and 21G needle, matrices were created in an oil-filled measuring cylinder, and this needle type was chosen for its flat tip, allowing for the creation of uniform spherical matrices. And the size and shape of the needle ensure that the size of form and droplets is suitable for our measurements, leading to more reliable and reproducible data. The quality of the obtained absorption and luminescence spectra was examined for the case where the slide was replaced with a thin flat mirror and for situations when the sphere remained in the Eppendorf or lay on black matte paper. And the least contaminated absorption spectra with the highest number of photons measured for a given wavelength were obtained for the measurement system in which the sphere was placed without an Eppendorf on a flat mirror. Here are 6 plots that represent data from measurements for 3 biopolymers and selected photosensitizers. And based on them, quantum efficiency can be measured. As you see, for protoporphyrin 9, gelatin matrices had much bigger absorbance than starch and agar. And despite that, luminescence is the lowest. Different situation can be observed with matrices doped with chlorine E6 and ferroforbit A. Absorption spectra is similar, but gelatin peak was wider. Also, the highest luminescence peak, but it's relocated towards lower wavelength. Agar matrices have had the most accurate luminescence and absorbance spectra, also connected with literature one, and have relatively high quantum efficiency, so they are the best for tested photosensitizers. On the previous slide, there was plots for only 3 photosensitizers because 5-aminolevulinic acid is a precursor that needs to undergo metabolic conversion to protoporphyrin 9 to exhibit luminescence. In our experimental setup, the lack of a metabolic environment means that 5-aminolevulinic acid did not convert to its luminescence form, resulting in no observable luminescence. This outcome was expected as the conversion process typically requires cellular metabolism, which was not presented in our in vitro setup. And the best spectroscopic properties were demonstrated by agar matrices, especially those impregnated with chlorine E6, which had the highest quantum efficiency among the tested photosensitizers, and gelatin influenced the expansion of the main band maxima for other photosensitizers. This could indicate stronger interaction or aggregation of the photosensitizers within the gelatin matrix, potentially affecting their efficiency and stability. And understanding these interactions is crucial for optimizing the delivery system to ensure consistent and effective therapeutic outcomes. And thank you for your attention.
Thank you for your presentation. And now it's time for question and discussion.
Thank you for a very interesting presentation. Could you tell us, because it was a preliminary study, what will be the next step of your study?
The next step of our study should be working with a line of cancer cells and to see how our matrices are behaving in a more neutral environment and see how their stability, and also it should be done in the future to observe how the velocity of dissolving these matrices will react in a more neutral environment, like, for example, testing with animals.
Thank you. Could you tell us what was your contribution to the study?
Of course, my contribution was to prepare half of these matrices. I've been doing this doped with ferroforbit A and chlorine E6. And I have been doing this, and I've designed the measurement system and watched also what would be the best solution to see the least contaminated spectra. So yes, that was my contribution to this project.
And okay, and you've prepared the those organic matters, and there was a starch, agar, yes, and gelatin, and then you said there was a tapioca. So the tapioca was the same as the starch, because the tapioca is from the manioc or something. Yeah.
Yes. Yes, of course.
That's the same.
Starch is like the substance in tapioca, so it was only a shortcut for me. But Yes, it was tapioca with starch in it.
Okay, thank you.
Thank you for the discussion. And now I would like to ask Marta Händler to present the paper Change in pulse waveform of cerebral blood flow velocity and total hemoglobin concentration in hypercapnia.
Okay, hello. Can you hear me well and see me well as well?
Well,
sorry, I didn't get that, but I think I am audible then.
Yes, yes.
I can hear you.
Okay, so let me share my screen.
Can you share your screen with presentation?
Yes, exactly.
Thank you.
Just a second. Is it good?
This is perfect.
Okay, thank you very much. Let me begin. Hello to everyone. My name is Marta Händler, and I'm happy to be able to present today a study performed by Arkadiusz Ziółkowski and me, which aimed to investigate temporal relationships between cerebral blood flow velocity and total hemoglobin concentration in the state of hypercapnia. Our study was in alignment with current trends in brain hemodynamics monitoring, which focuses on non-invasive measurement methods. And detailed analysis was possible due to high-frequency recording, which actually allows to inspect pulse-related waveforms and the signals coming from the brain. And also, since modern signal processing techniques are quite advanced, yet still fairly easy to use, we decided to investigate temporal rather than phase relationships. Firstly, I'd like to introduce the experiment setup and the measured signals. So the main signals were cerebral blood flow velocity, FV in short, measured by transcranial Doppler ultrasonography in the middle cerebral artery. And the second one was total hemoglobin concentration, THP in short, measured by near-infrared spectroscopy, functional near-infrared spectroscopy, NIRS in short, and it was measured on the surface of the head. So here on the photo, we can see a TCD probe here in the red square, and also the whole NIRS measurement cap with the channel which was later selected for analysis, which is highlighted in the light blue. Additionally, measured signals were arterial blood pressure, so ABP, and also end-tidal carbon dioxide pressure in the exhaled air, which is ETCO2. And all those signals were measured continuously and, like I said, non-invasively, and data was acquired using software called ICM+. The study consisted of 24 healthy volunteers aged 21 to 35, 7 of whom were males. And the hypercapnia test was conducted as follows. So first 5 minutes of measurements were performed in a state of normal capnia, which assumes a normal level of ETCO2, and it is basically the resting state. And after this period, volunteers were given breathing masks like the one in the photo here, and they were asked to breathe through it until, firstly, ETCO2 rose by around 10 millimeters of mercury, or until it reached a constant level. And then they continued to breathe through this mask in this setup for the next 5 minutes. So here on the plot on the left, we can see an example of acquired raw signals. So periods of normal capnia and hypercapnia are marked with gray and red, respectively. And on the right, however, we can observe those pulse-related waveforms that were present or are present in the signals after they have been filtered and detrended. And what we notice is that in both FV and THB, the signal morphology changes drastically between the states of normal and hypercapnia. Then data was processed as follows. So firstly, we located every pulse onset, which is simply a beginning of a pulse waveform. And then we split the whole signal according to those located pulse onsets to obtain single waveforms. Then for each waveforms, two variables were calculated. First of them was diastolic time delay, which is calculated as an absolute time difference between the onset of FV pulse waveform and the onset of THB pulse waveform. And the second variable was cerebrovascular resistance, CVR, which is calculated as mean ABP divided by mean FV. And the mean is calculated for the whole period of pulse waveform. These variables were calculated as absolute values, but also as relative difference between normal and hypercapnia using this simple formula. So those relative differences tell actually about the change in the variable in the state of normal hypercapnia with regard to the state of normal capnia. On this plot, there are measured signals in the three first subplots, along with calculated variables in the last two subplots for one of the volunteers. And there's a 10-second moving average applied to the signals and the 5-second moving average applied to the variables. Also, the dots on the lower subplots represent values without any averaging. And it is visible that both signals and also those variables change regarding their value when hypercapnia state is reached. So they change in this red zone. And to compare these values between the states of normal and hypercapnia, all values were grouped by volunteer and then by period. And also the median value was calculated for each volunteer and each period. So in this plot, there is a relative diastolic time delay for each volunteer. It can be seen that for the most of the volunteers, this value decreased in hypercapnia with regard to normal capnia, because our black bars representing hypercapnia are usually lower than the values for normal capnia, which are gray bars. And we can also observe that for several volunteers, like for example, N24 or N27, relative diastolic time delay remain at the same level, which is here denoted as 100%, because this value is the same as in the period of normal capnia. And the statistical significance of this change of diastolic time delay was confirmed using Wilcoxon test. And secondly, the correlation between diastolic time delay and CVR was calculated, and we used Spearman rank test. And overall, diastolic time delay was found to be positively correlated with CVR. On this plot, there is a clear positive correlation between change in diastolic time delay.
Sorry,
and
can you jump to the conclusion, because 7 minutes just passed?
Yes, I'm getting there. Yes, so the correlation coefficient was really high because it was over 0.5. And all correlation results were found to be statistically significant. So to conclude, the results confirm that the time delay between FV and THB pulse onsets gets shorter in hypercapnia, and this shorter time delay reflects also changes in the cerebrovascular resistance. Thank you.
Thank you very much for the wonderful presentation. And now I would like to open the discussion. Is there any question for our participants?
Thank you for your presentation. Could you tell us what's your contribution?
Yes, of course. So regarding my own contribution, I took care of the whole processing and calculation part. So once the signals were obtained, and they were obtained thanks to the work of Arkadiusz Wilkowski, I did the whole processing and the calculus, also with statistical analysis that was needed for this.
Please tell me also, I am interested how your result may be used or maybe may have impact on medicine in future.
Yes, so basically this work that I've started since I'm a first-year PhD student, this is a part of well, let's say bigger picture. So it is related to the future work that I would like to perform, and everything is in this alignment with the concept of non-invasive monitoring of brain hemodynamics rather than invasive. So everything that I try to do or analyze and everything that we undertake in Brain Lab is focused on searching for such measures or such ways to assess the state of brain hemodynamics, so that nothing invasive is really needed. So non-invasive measurements are sufficient to define the state of those brain hemodynamics.
But it's in patient with a stroke or
sorry,
what kind of patient we can use it?
Oh, well, I mean, the most well, let's say typical analysis are for the patients with hydrocephalus, and for example with subarachnoid hemorrhage. Yes, not particularly with strokes.
Okay, thank you.
Thank you very much for the discussion. Thank you once again. And now I would like to invite Teresa Kazimierczak from Wrocław University of Environmental and Life Science with the title Biological Activity of Polyphenolic Extract from Roots of Japanese Knotweed Rheinutra japonica.
Hello, am I heard? Everything is fine with my voice?
Yes, we can hear you.
Let me have a moment. I will share with you my screen. Can you see the presentation?
Yes, we can see.
Okay. Maybe I would like to stop this somehow. Oh, okay. Good morning. My name is Teresa Kazimierczak, and I am representing the Department of Physics and Biophysics of the Wrocław of Environmental and Life Sciences. And today I'd like to tell something about the biological activity of polyphenolic extract from root of Japanese knotweed. Before I will start to share my results, I will tell you something about the Japanese knotweed, because it's a common plant mostly native to the Far East regions, such as China, Japan, or South Asia, but it is also well known here in Poland. And it is known in Chinese medicine for its medical benefits, because it's rich in many substances that can be a treatment in many diseases, such as cardiovascular diseases, neuronal diseases, but also cancer. And what is interesting, it is known in Japanese cuisine as itadori. The aim of our study was to investigate the composition and biological activity of phenolic extracts of root of Japanese knotweed on the erythrocytes. We used the following techniques. First, we purified and analyzed the extract using mass spectrometry. Then we checked the phenolic content of the extract using Folin-Chokalteau assay. Then we started the measurements on the erythrocytes. First, we checked if this extract is toxic and if it's changed the osmotic resistance of the erythrocytes based on the release of hemoglobin, which was monitored spectrophotometrically. Then we checked the protection, if the extract protects the erythrocytes membrane against oxidative stress, and we used here fluorimetry and spectrophotometry. We also checked if the extract have any impact on the shape of the erythrocytes. We observed the erythrocytes under the microscope, and at the last results, the last measurements were done based on the. We wanted to check if where the extract is localizing in the membrane, how it's changed the properties of the membrane using fluorimetry. So the results showed that the extract is rich in polyphenols, mostly procyanidins, catechins, and stilbenes, which has very high antioxidative effects. And the total phenolic content was about 450 micrograms per gallic acid equivalent. We also shown that the results showed that the extract changes the osmotic resistance of the erythrocytes. It makes them more resistant to the osmolarity changes in the osmolarity of the environment in such lower concentration of 50 micrograms per milliliter relative to the control source. The impact on the protective impact of the extract was checked based both on erythrocytes and erythrocytes membrane. And here we have the graph that illustrates the hemolysis inhibition of the extract in the function of the concentration. The oxidative stress was induced by AAPH, and we saw that the hemolysis was effectively inhibited by this extract, and the IC50 was only around 60 micrograms per milliliter. And here on the right, we have the results from on the erythrocytes ghosts, which are the membranes, and we shown that both the extract is effective both against oxidative stress from AAPH and UVC, and we can see that against UVC, the extract has much better effect than the standard antioxidant drops. We also saw that the extract in concentration of 50 micrograms per milliliter can induce the erythrocytes shape to be more echinocytic than discocytic. It means that the extract may localize at the surface of the cells. And the last, we saw that we wanted to check how the extract modified the membrane of the erythrocytes. And here we used two fluorescent probes, DPH, which localizes in the inner membrane, and TME DPH probe, which localizes around, let's say, at the level of the glycerol in the membrane. And we see that both DPH and TME DPH anisotropy hasn't been changed much, maybe in higher concentration the TME DPH probe. That means that the extract is not affecting the fluidity of the membrane. So in conclusion, we saw that the root extract of Japanese knotweed is not toxic to erythrocytes. It makes them more resistant to the changes in the environment osmolarity. It protects the erythrocytes and erythrocytes membrane against free radicals induced by physicochemical factors such as UVC and AAPH. And it induced echinocytes and had almost no impact on the fluidity of the erythrocytes membrane. So the Japanese the polyphenolic extract of root of Japanese knotweed may have potential health benefits, and because it probably interact with the outer membrane of the cells, it can protect the erythrocytes against environmental stress from the outside. So here are the image lists, and thank you so much for thank you so much.
Thank you so much, and I would like to open the discussion. Dear jury.
Thank you for your very interesting presentation. Could you tell us what's your contribution? And I have the next question, Maya, is because this plant is also very popular in Poland, I checked. And could you tell us what was the antioxidant activity this plant compared with other polyphenols? It was better or the same?
Okay, so my contribution, basically this study was made by other students, master students. My contribution to this study is not only it's mostly the calculations, the graphs, and also preparation of the publication based on the studies. But also I performed another study on this extract that is not concluded in this presentation. And the second question was about how the extract
antioxidant activity this plant compared with other polyphenols.
Basically, we also checked the other extracts, also from Japanese knotweed, but extracted from the leaves, and the root had better antioxidant activity than leaves. It's probably because the root contains more tannins, and tannins has many hydroxyl groups. So maybe it's because of that the antioxidant activity is based on this. But also procyanidins, there are also many hydroxyl groups that both extracts were very drastically between each other. So in conclusion, I think that's why the root, because the root contains more tannins and procyanidins than leaf of this plant, it's more antioxidant.
And how did you get that extract? It's available commercially, or
no? The plant was harvested from the Wrocław Medical University, and it was extracted at our university by special methods.
Thank you.
Thank you.
Thank you for the questions and answers. And now I would like to invite our next presenter, Maja Ducka, to present the paper Analysis of Properties of HPMC-based composites produced using additive manufacturing for medical applications.
Good morning, everyone. Can you hear me?
Yes, of course. Can you share your screen with presentation for us?
Yes.
Thank you.
Can you see it?
We can see.
Okay, thank you. Hello, my name is Maja Ducka. I'm from Faculty of Mechanical Engineering at Wrocław University of Science and Technology. My presentation today is nie jest na pełnym ekranie? Sorry. Yes, and my presentation is the analysis of properties of hydroxypropyl metal cellulose-based composites produced using additive manufacturing for medical applications. Hot melt extrusion is a well-known technology for pharmaceutical sector. It allows for production of a wide range of oral drug delivery systems and enhances solubility and permeability of active pharmaceutical ingredients. It is also being researched as method to create feedstock for additive manufacturing techniques. Hot melt extrusion can be used to create filaments with containing active ingredients for printing of drugs with customizable properties or personal implants with complex geometry. Fused deposition modeling or fused filament fabrication is one of additive manufacturing techniques where the product is created layer by layer using a 3D computer model. Here, the material is in form of a thin cylinder, which passes through the nozzle, which hits the material, so it's partially melted, and deposited on building platform. Because the material is partially melted, the consecutive layers are attached to each other. But due to this method of creation, the properties of the finished elements are anisotropic. And the aim of this study was to gain a better understanding of chosen material properties of hydroxypropyl metal cellulose composites, which were created using this hot melt extrusion technique, and to be used in additive manufacturing methods such as fused filament fabrication. Afiniso hydroxypropyl metal cellulose for hot melt extrusion by DuPont company was chosen as the carrier in this study. It's amorphous, biodegradable, water-soluble polymer based on cellulose with a wide thermal processing window. So it can be used without causing drug degradation. Its glass transition temperature is quite low, at about approximately 150 degrees Celsius. And a lower melt viscosity allows it to be used potentially without plasticizers in hot melt extrusion technique. Two active pharmaceutical ingredients were chosen, ibuprofen and paracetamol, due to their low cost and accessibility. Triethyl citrate was used as plasticizers in 3 of these samples. 6 blends were created using these materials, and they are shown on the top in the table on the top. Those blends were processed using hot metal melt extrusion and then injected into dog bone shaped samples for tensile testing. And for compression testing, cylinder shaped samples were created using fused filament fabrication technology. 2 different testing machines were used, which are shown on the pictures on the screen, and testing parameters were chosen in accordance to current standards. The tensile strength test was used to measure 3 parameters: tensile strength, elastic modulus, and elongation and break, which is presented here as relative to length of the sample before the test. Pure HPMC was noted to have the highest strength of all samples. Based on obtained results, it can be said that both API and plasticizer noticeably affected mechanical properties of the material. Ibuprofen and paracetamol both acted as a plasticizer for HPMC, lowering durability of the material while increasing its ductility. HPMC tech paracetamol blend was noted as possessing the highest elongation, while lowest tensile strength and lowest elastic modulus of all analyzed blends. Its elongation and break was almost 300% higher than that of pure HPMC, while its strength parameters were about 90% lower. The blend containing both tech and ibuprofen also showed a significantly lower elastic modulus and strength. As shown in figure 6, pure HPMC in red exhibits near brittle behavior, fracturing almost immediately after reaching plastic deformation, and has no easily distinguishable yield point. Blends containing either active ingredients or plasticizer were more ductile and deformed plastically. Their uneven curves, shown here, suggest that the samples were cracking while they were being stretched. Some brief strain hardening can be observed in both samples containing APIs, here and here. and blends containing both plasticizer and active ingredients behaved like elastomers. The curves are quite flat. There's no clear elastic region or yield point to be observed like it was in other samples. And they were searching very quickly under quite small load until they reached the breakpoint. Compression tests were additionally used to measure yield point and elasticity modulus of HPMC and active ingredient blends at different temperatures of the nozzle during printing of the samples. Blends containing paracetamol had higher compressive strength, and it can be noted that as the temperature of the nozzle increased, so did the value of the parameters, as can be seen on graph on figure 7 on the right. Nozzle temperature had bigger impact on the blend with paracetamol, where elastic modulus was almost 5 times higher between the highest and lowest temperature. And that indicates that only at higher temperatures material was properly plasticized, which allowed for creation of samples with a better structure. It signifies that only at high temperatures material was melted enough, so the consecutive layers were properly attached to each other.
I'm sorry, could you just jump to the conclusions? Oh,
yes,
thank you.
I'm reaching them. Sorry. So addition of tech and active ingredients significantly changed mechanical properties of the blend. The ability of the samples to carry loads was diminished, especially with those with both plasticizer and APIs. With such low tensile strength, the number of applications is reduced. It should also be mentioned that while higher nozzle temperatures improve strength parameters of the samples, APIs can be destabilized and start degrading in such high temperatures, leading to either lesser effect of the drug on the patient or creation of undesired waste. And although there is no official published set of appropriate parameters for FFF technique filament, in literature it's suggested that the elastic modulus should be between 300 and 700 megapascal, and such filament should be stiff enough to retain the required shape after being pressed out, but not be too brittle as to break in the nozzle. Printability of material is based on more parameters than this criterion, but it can be used to preliminarily eliminate some. Blends containing both API and plasticizer had modulus much lower than the recommended, and such high plasticity can lead to non-optimal fitting of the filament and clogging of the nozzle. Thank you.
Thank you for your presentation, and now I would like to open the discussion.
Thank you for your presentation. Could you tell us what your contribution was? And I have another question. Could you tell where your method might be used in pharmacy or medicine?
So I helped create the samples. I did all the tensile tests and then processed the data and analyzed it. And well, the currently used materials are can be used, but are not ideal. So we are still trying to find better and better materials for the use in pharmaceutical and medical field. And to be used, they have to be quite well researched. Proper parameters should be chosen, proper blends should be chosen. So here in our department, we have one of the projects is to analyze how these cellulose-based polymers react with other materials and how can they be prepared to be properly used. Because right now, as you can see, some parameters are too low to be used in load-bearing implants, which is our goal.
Thank you.
Thank you.
Thank you once again for your presentation. And now I would like to invite Agnieszka Prusek from Medical University of Silesia, Katowice, with presentation titled Expression of Genes Related to NLRP3 Pathway in benzalkonium chloride-treated limbal stem cells.
Good morning, ladies and gentlemen. I would like to present the work titled Expression of Genes Related to NLRP3 Pathway in benzalkonium chloride treated limbal stem cells. corneal integrity depends on the self-renewing properties of the corneal epithelium. This renewal relies on limbal epithelial stem cells. The lack of corneal regeneration due to the less dysfunction may negatively affect the condition of the cornea. Long-term administration of eye drops with preservatives may lead to dry eye disease, which is accompanied by inflammation, apoptosis, and local damage of the cornea epithelium, which constitute a vicious cycle of dry eye disease. Benzalkonium chloride is one of the most popular preservatives using in eye drops. To date, there is a lack of studies investigating back activity on human limbal epithelial stem cells population in vitro. The use of LPS as a strong pro-inflammatory agent is a well-known model of inflammation. Binding of LPS to TLR4 triggers a signaling cascade. This leads to the activation of transcription factor NF-kappa beta, which leads to the activation of NLRP3 pathway, and then activation of caspase-1, and the production of pro-inflammatory cytokines interleukin-1 beta and interleukin-18. MMP3 and MMP9 are also connected with inflammation. Tissue inhibitor of metalloproteinases 1 can inhibit MMP3 and MMP9. The aim of this study was to evaluate the impact of benzalkonium chloride on limbal stem cells in vitro. The experiments were conducted on human limbal epithelial stem cells isolated from corneoscleral rim. Then, in order to identify the RT-qPCR and immunofluorescence staining was performed. Lests were cultured for 48 hours in a back solution in dose 0.0002% and in LPS in concentration 2 micrograms per milliliters as a positive control. Then the expression of genes related to NLRP3 pathway and MMP activation was assessed. Lests were isolated by explant method. The expression of cytokeratins 3 and 12 was not detected both at gene and protein level. The intensive expression of P63 and ABCG2 suggests that isolated cells represent stem cells population. The expression of interleukin-1 beta was significantly increased, and the expression of caspase-1 and interleukin-18 was significantly decreased in back group compared to the control group. I did not detect significantly difference in NLRP3 gene expression in all groups. These results may suggest dysregulation of NLRP3 pathway by back, leading to reduced inflammatory activation, but increased interleukin-1 beta production, maybe through other alternative signaling pathways. The expression of caspase-1 and interleukin-18 was significantly increased, and the expression of interleukin-1 beta was significantly decreased in LPS group. Compared to back group, that this means that the potency of back is not as strong as LPS. I also noticed significantly increase in expression of caspase-1 in LPS group compared to control group. It's possible that despite high levels of caspase-1, there might be a delay or impairment in the processing of pro-interleukin-1 beta to the active form. This delay could result in the accumulation of pro-interleukin-1 beta or decrease in the secretion of mature interleukin-1 beta. The results clearly show activation of MMP3 and MMP9 by LPS. I did not detect statistically significant in expression of MMP3 and MMP9 between back group and control group. The expression of tissue inhibitor of metalloproteinases 1 was lower in back group and LPS group, but without significant differences. The reason why BAC does not directly active MMP3 and MMP9 may be attributed to its mechanism of action. In conclusion, the LPS induced inflammatory response is associated with the NLRP3 pathway and MMP activation. BAC indirectly affects the NLRP3 pathway but does not active MMP gene expression, but does not induce inflammatory response as strong as LPS. Thank you for your attention. If you have any question, I will be pleased to answer them.
Thank you for your great presentation. And now it's time for questions.
Could you tell us what was your contribution and are you going to continue your research? What will be the next step?
I did all research at laboratory. Then I analyze these results and I'm presenting this work here, but I always could rely on my PhD supervisors, and I'm really thankful for it. Next step, I plan to continue this research in the area of therapy of this toxicity model, because first step of my PhD plan was to working on back toxicity model on limbal epithelial stem cells, and then in the second step, I try to create new therapy on this disease.
Thank you for your speech, and I have one question. The BAC is the most common preservative in eye drops, but do you have some plans to compare with another preservatives, or only you focus on BAC?
I focus on benzalkonium chloride because it's the most popular and the most dangerous agent. But on the other side, it's still the most important problem of this preservative because I was talking with maybe other other scientists, and like I said, it's the most important problem because other preservatives are not that important.
Yeah,
like benzalkonium chloride. That's why no.
Okay, it's the most common, and this is a problem because it's a long-term therapy. So, so these preservatives affect and eye surface.
Yes, and especially in glaucoma eye drops. So.
Thank you for the discussion, and now. I would like to invite the next presenter, Al-Hassan Ali Ahmed.
Hello, everyone. Hello, professors.
Hello.
Do you hear me and see my screen?
Yes, we can hear you, but we don't see your presentation now. And can you? Oh, okay. Great.
So is it visible now?
Yes, it's visible on full screen. Thank you.
Okay. So, hello everyone. I am Al-Hassan Ahmed, a PhD candidate at Bosnia University of Medical Science, the Department of Bioinformatics and Computational Biology. It is an honor and pleasure to present my work here today. My topic is about artificial intelligence model in lung cancer diagnosis. under the supervision, of course, of Professor Elizabeth Kaczmarek. The presentation will include an introduction about cancer diagnosis and pathologist situation and concerning AI applications in Poland, methodology of our project, results of our model, including accuracy and leading time, and finally, the conclusion we achieved. As you may know, lung cancer is the most common cause of death from malignant tumors worldwide, with more than 2 million patients diagnosed yearly, while cancer is the second cause of death in Poland, with more than 100,000 deaths per year. Pathological diagnosis is a crucial part of cancer treatment. However, according to the latest study, the average time the patient takes from the first symptoms to diagnosed with cancer is almost 3 months. The main issue here is the number of active pathologists, which is too low to maintain a swift time for the proper cancer diagnosis. According to the latest report, there are less than 800 active pathologists, making the number of 2 active pathologists per 100,000 citizens one of the lowest European countries. Implementing AI applications in medical field could save time and money, in addition to reducing the error possibility and prolonged time of waiting by patients. Hence, our project aims to develop a machine learning model for efficient lung cancer detection, supporting pathologists in the diagnosis process of their daily basis. In order to have an excellent machine learning model starting from data input and going through image analysis to have the final output, whether it is cancer or not, you need to follow these steps. First of all, data collection involve, having high quality images or slides. Then you have the data pre-processing. We have techniques such as normalization, which gives a standard scale of 0 or 1 for each feature to assure better understanding of the model. And also we have the data augmentation, which increases the interpretability of the model by introducing different images to the model by, doing crop, rotate, flipping, resize, and color correction. Then we have feature selection, include texture, color, shape, and other properties that can help identifying tissues and cell types. The most crucial step, which is model selection, and we have also training and validation step. Then finally, testing and deployment step, which is testing the model performance using new data which were not used before in the training and validation step. We started our project in 2022 by conducting personal interviews to gather pathologist input toward AI application in medical diagnosis. A total of 68 pathologists from Poland participated in the study, which gives 15% of the total active pathologists in Poland. The average age of participating pathologist was almost 39 years old. 53% of the pathologists were female. The pathologists have an average of 12.5 years of medical experience. Almost 58% of them have no previous experience with AI or machine learning in general, and particularly in medical field. Moreover, on a scale of 10, pathologists expressed their trust in AI results with a mean of 6.3. Also, they reported the efficacy of AI in diagnosing cancer cell with a mean of 6.4. After gathering the pathologist opinion, we started the slide data collection, pre-processing, and designing our model. We obtained 170 whole slides stained with hematoxylin and eosine dye. Each slide was used to randomly extract an average of 6 images by implementing data augmentation techniques that I explained earlier. This process resulted in 934 images divided into 557 cancer and 377 healthy. We followed the known approach of 70% training data versus 30% testing and validation. We selected convolutional neural network model to operate our algorithm because it is specifically designed for image analysis task, taking advantage of the inherent spatial structure of the images. Our results, as shown in the table, model accuracy is more than 96% in the distinguishing between cancerous and healthy cell, lung cell images within 10 seconds. The model was validated by 10 active pathologists from the pulmonary department at the teaching hospital of Bosnia University of Medical Science. Each pathologist was provided with 15 images and asked to classify them as healthy or cancer or uncertain. The pathologist average accuracy was 79%, while our model overall performance exceeding active pathologist with an accuracy of 100%. This outstanding performance of our model is due to it was designed to handle complex nonlinear relationships, fault tolerance, and pay attention to the true positive. Therefore, it can capture and detect fine-tuned details. Eventually, it's worth mentioning that staining patterns were not one of the criteria the model used in making decision to avoid false positive and false negative issues. Here, as you can see in this demonstration video, firstly, I have to upload the image I am planning to check. Then the model gives either a score of 1 or 0 based on the analysis. 1 equal cancer, while 0 is for healthy. Then the model reprocesses the image itself in order to interpret the results and highlight the areas where the decision was made of. As you see, we have 6 places where the model finds cancerous cell within the specified time. In conclusion, we designed our model based on optimized residual blocks to enhance feature representation without compromising detection accuracy. That's why the accuracy is more than 96%, which is great in the artificial intelligence and machine learning field. Of course, within a swift time of 10 seconds. The model outperformed results reported by active pathologists, suggesting that AI algorithm assisted by pathologists can enhance diagnostic skills and reduce leading time in diagnosing process. Universities, healthcare facilities can use our model to train medical students and junior physicians because the accuracy of pathologists has improved over their years of experience. Also, it was clear that pathologists require more training in order to better understand and use AI tools in medical diagnosis. Finally, I would like to announce that our project article was published last month in Cancer Journal with our results and validation data. Thank you all for attention, and please, if you have any questions, feel free to ask.
Thank you for your presentation, and now it's time to ask a question.
So thank you for your presentation. It was really nice. I have a question. Have you tried already to implantation in the clinical field? Have you ever worked with any clinical pathologist?
Yes, we already started to use them with this 10 pathologists who involved in the validation process and the in the Poznan department, pulmonary department at the teaching hospital of Poznan University. Also, it's still not easy to generalize this model in all clinics, but we started already with this 10 pathologists, and the results, as explained, it was really great.
Okay, and only for the lung cancer, or have you tried any other adenocarcinomas?
As you see,
GI
Professor,
tracts?
Yeah, as you see, Professor, there is a lot of potential. So, for example, the future, our future plans is to generalize it for different type of cancers, to also start to improve the model to be able to grading the cancers, like, which grade on which stage, also to start to optimize the treatment. So there is a lot of potential coming out from this model, and we can use, of course, to collaborate with the specialized people in order to have, a unified and conclusive data.
Thank you. Could you tell what was your contribution?
So almost everything, Professor. So the idea was mine, the concept, methodology, also involved in doing these interviews with pathologists, but of course with assistant from someone who could, translate and using the Polish language, of course. The validation process, because also it was not only validated by the pathologist, but also we used outsourced data from Dartmouth University in United Kingdom. We asked them to provide us with more than 1,000 slides to validate also our model, and it was great. Also, as I mentioned, writing the manuscript and doing the analysis and showing the results. So let's say 90% of the project was by me.
Thank you.
Thank you so much for your presentation. And now I am glad to invite Aleksandra Gładyś from Medical University of Silesia with a presentation titled Procancerous Effect of Adipose-Derived Mesenchymal Stem Cell Medium in Hepatocellular Carcinoma.
Okay, thank you. So, ladies and gentlemen, I have an honor to represent the Department of Histology and Embryology at the Medical University of Silesia and present you the results of my research on liver cancer and its interactions with stem cells derived from fat. And to be more precise, I am investigating hepatocellular carcinoma, which is the most common cancer of the liver, and human adipose-derived mesenchymal stem cells, which are the population of cells physiologically responsible for restoring cell population in fat and the regeneration of this tissue. And besides that, research suggests that although they have this physiological function of restoring the population, they may have also an anti-tumor activity in certain types of cancer because of the substances that are produced by these cells, especially prostaglandin E2 or transforming growth factor beta or interleukins may have this anti-proliferative and pro-apoptotic effect on certain cancer cells. And hepatocellular carcinoma is distinct among other cancers in such a way that their proliferation strongly relies on the Ras-Raf-MEK-ERK signaling pathway. And this pathway is a subject of extensive research in oncology and is a target pathway of many drugs used in this disease, such as sorafenib or regorafenib. And the use of these drugs prolongs this progression-free survival in patients and also overall survival because it halts the proliferation of the tumor. And here I get to the aim of my study, which was to determine if we can help these drugs and specifically what stem cells can add to these standard therapies to enhance this anti-cancer effect in this disease. And what we have done was to collect culture medium from adipose-derived mesenchymal stem cells, which were previously characterized by flow cytometry markers, and their potential, the differentiation potential was evaluated as truly mesenchymal. And then I used this medium to culture it with HepG2 hepatocellular carcinoma cell line, with or without the presence of sorafenib in culture media. So this one of these drugs which are standardly used in this disease. And after 2 days of culture, I evaluated the viability of the cancer cells, the apoptosis, alpha fetoprotein gene and protein expression, and the expression of genes of this Ras-Raf-MEK-ERK signaling pathway I mentioned before. So basically I had 4 groups. Try with this one. Okay. So one was truly the control group. So HepG2 cells cultured in standard medium. Then I used, the second group was with this conditioned medium. I think the pointer isn't working, but I can point it that way. The third one was the group with sorafenib, 7.5 micromoles of sorafenib, and the last one was with both. So both with sorafenib and this conditioned medium from adipose-derived mesenchymal stem cells. So let's go quickly to the results. So viability was measured with an MTT test, and as we can see in this second bar, so the use of sorafenib in both of these groups, with sorafenib, the viability is decreased, and at the same time, the use of conditioned medium increases the viability of the cells. And in the last group, when we used both of them, we see that the effect of sorafenib is stronger, so the viability is decreased in the end. The second step was to evaluate pro-apoptotic genes. So BAX, Caspase-3, Caspase-7, and TP53, they are all pro-apoptotic genes, when the expression is higher when the cell is getting into apoptotic pathway. And the use both of sorafenib and conditioned medium have to some extent, a pro-apoptotic effect, but we couldn't prove that one is better than the other or the use of both is more beneficial than the other one. So to prove it with another method, I use flow cytometry analysis with annexin 5 and propium iodine. And here we can see that the use of sorafenib causes a shift from viable cells into early and late apoptotic cells, so it's the second bar and the last bar. And the use of conditioned medium slightly increases, but it wasn't statistically significant in that case. But the difference between sorafenib groups and conditioned medium group is also significant when compared them to each other. And here are the same data, but shown in different ways. So it's pretty much the same data used in here, but without marks to be more clear. And here we can see that the percentage of early and late apoptotic cells in groups with sorafenib are higher. And next step was to evaluate the gene expression of Rasov-McErk pathway, so the pathway that causes proliferation in these cancer cells, and the expression of genes connected to this pathway are higher, mostly in the group with conditioned medium. And the last step was alpha-fetoprotein expression, which is the marker of hepatocellular carcinoma cells, and is higher when the tumor mass is higher, and the use of conditioned medium increased the expression, and the use of sorafenib decreased the expression and IFP, but it wasn't visible clearly on protein level used by with immunofluorescence. And jumping into the conclusion, the increase in IFP expression and cell viability after incubation with conditioned medium suggests that the medium has a pro-cancerous effect on HepG2. So, however, certain pro-apoptotic effect was also visible in some cases, and this research suggests that the conditioned medium and sorafenib, they have rather opposing effect rather than synergistic, as I hypothesize about. Thank you.
Thank you very much for a great presentation and quickly conclusions. Maybe we have any questions?
Could you tell what was your contribution and did you receive any funding for your research?
Yes, I received the funding from Parts of Science from the Ministry of Higher Education. And my contribution was I did all the cell culturing, and then I made most of the tests alone, but some of them I was assisted with my colleagues. So I did an MTT test by myself, and also RT-qPCR I did myself. Immunofluorescence I was assisted by Dr. Bartosz Sikora, and with flow cytometry I was assisted with Dr. Patrycja Wieczorek.
Thank you.
Do you use these results to your thesis? It's your thesis project?
Yes, it's half of my thesis, yes.
Thank you.
Thank you very much. And now I would like to invite Gabriela Orzeł to present the presentation, Classification of Heart Projection in Transthoracic Echocardiography using Artificial Intelligence Methods. Okay, so if Gabriela Orzeł doesn't hear, we can move this presentation on the end of the session. And now I invite the next presenter. With presentation, uric acid reduces telomere length, and Mendelian randomization study.
Hello, can you hear me? Okay. Am I visible?
Oh, Karol Krystek, the floor is yours.
Okay, thank you very much. Good morning. My name is Karol Krystek, and I'm more than happy to be here today and present you my work titled Uric Acid Reduces Telomere Length, a Mendelian Randomization Study. So without further ado, let's move on to the content of my presentation. Firstly, I will give you a short introduction regarding oxidative stress, uric acid, and Mendelian randomization. Secondly, we will move on to the aim of the study, followed by material, methods, and results. Lastly, I will shortly conclude obtained results. So what actually oxidative stress is? Oxidative stress is a physiological imbalance between the production of reactive oxygen species and the body's ability to detoxify or repair the resulting damage. ROS are highly reactive molecules containing oxygen, which are produced as byproducts of normal cellular metabolism, particularly in the mitochondria. Under normal conditions, the body has antioxidant defenses mechanisms to neutralize ROS and prevent cellular damage. Antioxidants, including enzymes as well as non-enzymatic molecules, work together to counteract the harmful effect of ROS. However, when ROS production exceeds the body antioxidant capacity, oxidative stress occurs. This can lead to damage to lipids, proteins, and DNA within cells, disrupting cellular function and contributing to the development of various diseases and conditions, including aging, neurodegenerative diseases, cardiovascular diseases, cancer, or diabetes. So what connects oxidative stress and uric acid? Uric acid is a heterocyclic compound that plays important roles in the human body, particularly in the context of metabolism and disease. It is a waste product formed during the breakdown of purines, which are substances found in many foods and also formed naturally by the body. What is more important, uric acid exerts both pro- and antioxidant properties. Uric acid is one of the major endogenous antioxidants in the human body, scavenging free radicals and reactive oxygen species to prevent oxidative damage to cells and tissues. In conditions of moderate oxidative stress, uric acid can exert protective effects by neutralizing ROS and mitigating oxidative damage. However, under certain conditions, elevated levels of uric acid can promote oxidative stress and contribute to cellular damage. High concentration of uric acid may lead to generation of ROS. This can amplify oxidative stress and contribute to the shortening of telomeres, which leads to pathogenesis of various diseases. The question is, can you hear me? The question is how to prove that increased uric acid levels shorten telomeres. Hopefully, Mendelian randomization comes to the rescue. And Mendelian randomization, MR, is a statistical technique used in epidemiology and genetics to assess actual relationships between risk factor and an outcome, typically in observational studies. The method relies on the genetic variants that are associated with the risk factor of interest but are not influenced by the outcome. These genetic variants act as instrumental variables akin to randomized control trial in which individuals are randomly assigned to different levels of the risk factor. The key assumptions of MR is that the genetic variants used as instrumental variables affect the outcome solely through their association with the risk factor and not through alternative pathways. I will try to explain the assumptions of this method based on this diagram. Firstly, we need to identify genetic variants, in this case, single nucleotide polymorphism, associated with uric acid levels. These genetic variants are randomly distributed during meiosis and are not influenced by telomere length. Similarly, genetic variants associated with telomere length are selected. It is important that no confounders such as population stratification, gene-environment correlation, horizontal pleiotropy, reverse causation, and measurement error, or small sample size are associated with the genetic instrument. And secondly, that the genetic proxy of exposure, SNP, should not be independently associated with the disease outcome. Exposure and outcome are categorized into groups or as continuous variable. So what was the aim of my study? The aim of the study was to demonstrate the influence of genetically predicted uric acid levels on telomere length and vice versa using the Mendelian randomization. To achieve that, AMR was utilized to determine the impact of genetically predicted UA concentration on TL and of genetically predicted TL on UA concentration. Inverse variance weighted mean approach was used in primary analysis. Secondary and sensitivity analysis included Cochrane Q test, leave one out analysis, MR Egger, and MR Presso. Data on single nucleotide polymorphisms affecting UA concentrations and telomere length were obtained from UK Biobank for nearly 500,000 samples each. Instrumental viable SNPs were selected based on previously reported associations with telomere length and from genes responsible for UA synthesis and transport. The obtained results present as follows. A total of 12 SNPs were included in the analysis of UA levels, impact on telomere length as well, and 14 SNPs in the analysis of telomere length impact on uric acid levels. And the increased UA concentrations were found to significantly reduce telomere length. In all three tests, p-value was below 0.05. Whereas telomere length did not affect UA levels at all, the p-value was above 0.05. What's more, no significant horizontal pleiotrophy was detected in either analysis by MR Egger intercept test and MR Presso, and findings were not affected by a single SNPs in leave one out analysis. I will try to prove it with those graphs. Both graphs represent the impact of instrumental variable SNPs on telomere length and uric acid levels. Each dot represents a single SNP, and lines represent 95% confidence interval. Trend lines represent causal estimates of different MR methods. The trend lines on the left graph are not significantly different from zero, therefore telomere length does not affect uric acid levels. However, the trend lines on the right graph are significantly different from zero and differ significantly from zero after averaging with weighted median, MR Egger method, and inverse variance weighted median. Now, to sum up, this MR analysis showed a causal link between uric acid plasma levels and telomere length. These results showed the possibility of using this causal link as genetic and screening tests and prognostic markers for diseases related, for example, to oxidative stress. But the exact mechanism remains to be elucidated. There are some of my references. And thank you very much for your attention. And if you have any questions, I will be more than happy to answer them.
Thank you for your presentation. And thank you once again for your presentation. And now I would like to open the discussion.
Thank you for your presentation. Could you tell us what your observation is? Because different guidelines say that the patient with, especially patient with cardiovascular diseases, the level of uric acid should be below 5 and should be very low. What the observation is good because you show that uric acid has also antioxidant properties.
Yes, it was worth noting that in this analysis, the SNPs of uric acid levels was associated mainly with the transport of uric acid rather than synthesizing it. So it is it's hard to tell whether lowering the synthesis of uric acid endogenously will affect will affect the telomere length in the same way.
Thank you. What's your contribution? Because could you tell?
Yes, so I took part in almost every step of this analysis, from verifying whether those SNPs should be included or excluded from the analysis. I were searching the UK Biobank for the data obtained for previous experimental studies, and then I took part in statistical analysis of this causal link and correlation between uric acid and telomere length.
Thank you.
Thank you once again for your presentation. And now I would like to invite Maciej Borówka from Medical University of Silesia. Trust is good, but in medicine control is better. How much natrium butyrate is in natrium butyrate products?
Good morning, ladies and gentlemen. I have a great honor and pleasure to present to you on behalf of my colleague Michał Nicze and me our work on how much natrium butyrate is in natrium butyrate products. In our study, we compared 8 different products registered as dietary supplements containing natrium butyrate. All 5 stages of this experiment were carried out in laboratories of Department of Pharmacology in Katowice, entirely by ourselves. Here I present to you plan of my speech. To begin with, natrium butyric acid is a short-chain fatty acid produced by bacteria in digestive tract. It serves mainly as a source of energy for colonocytes. Its derivative natrium butyrate is a popular diet supplement used in many conditions. According to producers' recommendations, many general practitioners and gastroenterologists advise patients using them in conditions such as irritable bowel syndrome or inflammatory bowel diseases. We also observe that in our clinical practice, in patients admitted to Department of Internal Medicine we work in. But the point is that all products containing natrium butyrate are registered as dietary supplements, not medications. These terms are defined by Polish pharmaceutical law. We summarized main differences between them in this table. In short, medications are associated with much stricter regulations. They need to be controlled continuously. In our study, we compared 8 different products. We summarized main differences between them in this table. As you can see, 7 out of 8 were in capsule form, and also they differed in terms of dosing and containing excipients. In our study, we used methods previously described in literature. It was methods for quantitative determination of short-chain fatty acid in biological materials using high-performance liquid chromatography. We started with crushing tablets and contents of capsules in the mortar. Then we put the powder in test tubes, added strictly defined amount of propionic acid as an internal standard, and the ethyl ether to extract short-chain fatty acids. If you are wondering, this hand is mine. Then we performed vortexing and centrifugation. After that, we evaporated the ethyl ether. And then remnants were dissolved in methanol and put in ultrasonic bath. Then we used sterile syringe filter to filter the samples and put them in vials appropriate for autosampler of chromatograph. Now let me tell you a few words about high-performance liquid chromatography. It's a well-known method used for determination of amount of some substances such as drugs in serum or plasma. To begin chromatography, we installed a chromatographic column in the machine and set parameters such as temperature, time of procedure, and flow of solution. Here we present you obtained chromatograms. On this axis, there is time, and this peak corresponds with analyzed substances. This one is propionic acid, and this is butyric acid. They are separated based on their molar mass. When the substance exits the column, reaches the detector, we used ultraviolet detector. And when it reaches the detector, the peak comes. The higher the peak, the higher the concentration of the analyzed product. We also prepared exemplificatory solutions of propionic and butyric acid to prepare calibration curve. Here we present you our final results. As you can see, all of these substances contained natrium butyrate, and the percentage of declared content varied from 40 to more than 100% of declared content. We also summarize that in this chart. As you can see, 5 out of 8 had really similar content to the declared content by the producers. However, our study had some limitations. For example, the method was primarily designed for using in biological materials, and excipients present in analyzed products might have influenced final results. In conclusion, extraction with diethyl ether and high-performance liquid chromatography might be an effective method for determining of concentration of butyric acid in various materials. Analyzed product did contain natrium butyrate, but they varied in terms of percentage of declared content from 40 to over 100%. As for conflict of interest, we don't have any to declare. Thank you for your attention. Now we'll be pleased to answer any questions you may have.
Thank you for your presentation. And now I invite to the discussion with our authors.
Thank you for your presentation. Could you tell us what was the key of choosing this particular supplements for your analysis?
Thank you for the question. It may seem they were chosen totally randomly, but the selection criteria were based on the both clinical and epidemiological factors. I mean, first of all, we observed and analyzed the natrium butyrate supplements the patients admitted to our clinical department took. And then we conducted a kind of a survey among some local pharmacies and asked the workers which natrium butyrate supplements were bought most frequently by people. Then we purchased on our own 8 out of many available supplements, and we analyzed them in the laboratory. We intended to combine both laboratory experimental background together with some clinical future applications.
If I may add, patients also use trade names of the substances, so it was easier because of that. They don't tell us they take natrium butyrate, they tell and they use names. I can't.
What's your contribution? Standard question for each presenter.
All steps of the laboratory test we performed by ourselves. In planning the experiment, we used help of our tutors, and they helped us sum up everything.
It's your PhD thesis or not? It's only study.
It's connected, but it's not exactly the topic. We were checking if the high-performance liquid chromatography is, if can we use it for determination of concentration of short-chain fatty acid in biological material of our patients, and we decided to start with something simple. So we thought that drugs will be simple and that we can also use that in our clinical practice.
Thank you.
It's very interesting. Thank you. I have one question. Were you surprised with your results or no? About this content?
To some extent, I think we were surprised because people think that supplements are registered as this in the same way as the popular medications, and they took these products thinking about they are the same as the medication So to some extent, we were surprised. We were thinking that most of these products wouldn't be comparable to the amount we could measure. But as it turned out, a majority of them, I mean 5 out of 8, were comparable. So it means that the producers which manufacture these products put a proper amount of this natrium butyrate in these tablets or capsules. So,
and if I may add, when we were performing laboratory tests, we felt the characteristical smell of butyric acid. So it wasn't so much a surprise at the end, because we know that they really have that, but we didn't know how much exactly.
Yeah.
Thank you once again. And now we would like to invite Gabriela Ręka with a presentation titled the analysis of variants of unknown clinical significance from NGS results in children with autism.
Good morning. Do you hear me?
Yes, we hear you.
And I'm trying with my present. Do you can you see my presentation?
Not yet.
So maybe we can wait, because I see that it is.
Okay, and can you change it for full screen?
Okay. Yes. Can you see it now?
Not in full. Oh, just a second. No, it's.
I can see that it is visible.
Oh, yeah.
My screen.
Now it's perfect. You can start if you're ready.
You. So once again, my name is Gabriela Ręka. I represent independent diagnostics of the second department of pediatrics from Medical University of Lublin. And I have a great pleasure to present you today my work about variants of unknown next generation sequencing results in children with autism. Autism spectrum disorder is a group of complex neurodevelopmental disorders that include 3 components: deficits in social communication, in maintaining relationships, and repetitive atypical behaviors. And among them are several reasons, like prenatal, perinatal factors, genetic, and some immunological or environmental factors, also some neurological factors, and even gastrointestinal factors. And it is in the literature, it is said that about 1/3 of cases of autism can have a genetic reason. And among those genetic changes, copy number variants were found, inversions, translocations, single nucleotide variants, and point mutations. And they were found on different chromosomes. And it is said that more than 1,000 human genes are associated in different ways with autism. And in next generation sequencing, NGS, it is technology of sequencing that can enable to sequence different genes. And this method might detect point mutations and suggest copy number variants. And it also can evaluate mitochondrial DNA. And the aim of my study was to analyze sequence of DNA from NGS in patients with autism, to compare the changes between patients, assess genotype and phenotype correlation, and overall research of genetic causes of autism. And in my research group, I included only pediatric patients below 18 years old with diagnosis of autism, also Asperger's syndrome, and those who were diagnosed with NGS The study was approved by the Bioethics Committee of my university, and it was retrospective and prospective study. I included children who were diagnosed from 2017 to 2023 in genetic outpatient clinic in University Children's Hospital from Lublin. This NGS was performed as whole exome sequencing or next generation sequencing panels. So genes connected with ASD and possible other diseases of the patient. And I analyzed also patient's medical documentation. And in the research group, the majority of the NGS tests were panel tests. I included 73 patients. Mean age was 8.5 years old. And all patients came from Poland. The majority of patients were boys. And I had one sibling pairs. And in my group, 65% presented dysmorphic features, face dysmorphic features, and half of the patients presented disability. And in my group, few patients were adopted, so I do not have full family history. And neither of parents of patients had autism. However, about 10% of siblings of my patients had also autism. And this about it, normal karyotype was in about 50% of patients. In microarrays, normal results were in 64%. And others had duplications, deletions, or multiple copy number variants. And here, please look at pathogenic variants. 72% of patients had pathogenic variants in NGS, but my work is about variants of unknown significance. So this here is pathogenic variants connected with autism. So 54%. And here you can see this variants of unknown significance. So these are variants which we do not know now if they are pathogenic or they are mild. And here I show at the table in the left variants repeated in 2 adults or in more than 2 patients. And in the right, I put variants only in single patient. So we can see that some of them are repeated. And here in the tables, I present you those repeated variants. And for the function of this gene, and possible disease that can be seen in patient due to this mutation. And I in red, autism is one of a symptom. So you can see that even some of detailed results are repeated.
I'm sorry to interrupt, dear Gabriela. Sorry.
In 4 patients, some on 2 patients. You can see generally how many.
Przepraszam, że przerywam. Halo?
Hello?
Mamy bardzo problem z połączeniem. Właśnie mamy problem z połączeniem. Bardzo nam panią przerywa. Może to być wynikać, nie wiem, z połączenia z internetem.
I'm sorry, I'm maybe my Zoom, but to sum up, I would like to say that most of this mutation, however, some other type of point mutations like frame shift, and 62% of these variants were possibly connected to autism. So to conclude, autism in most patients results from other than genetic reasons. Some patients present with visible genetic reasons due to different mutations connected with autism. And the study indicated great heterogeneity of variants of unknown significance. All patients should have NGS testing performed, patients with ASD. And because of such amount of repeated variants of evaluating databases used to analyze NGS results and possibly change some variants classifications. Here I present you bibliography. Thank you very much for the attention. And do not hesitate to ask questions.
Thank you for your presentation. And now I will open the discussion.
Can you hear me?
Yes, we hear you.
Does do scientific committee or any of participants have questions to me?
Yes, we have some questions. First of all, because we have some hearing problems during your presentation.
I'm sorry, maybe because of unstable internet connection.
Yeah, okay. But could you tell us what was your contribution into presented work?
Okay, so I gather these results. So I chose patients with autism from this medical documentation, and I analyzed these results. So it is my contribution because I personally did not perform these tests, but I analyzed the results and gather it.
Okay, and those tests was performed commercially, or did you have any grant for that?
No, I did not have grant. These tests were performed during standard diagnostic in genetic outpatient clinic in the past. So I used these results for my analysis.
Okay, and do you know if any patient with ASD has the genetic testing?
I can repeat. Can you repeat this question?
For patient with the ASD, they have the genetic testing during the diagnostic process.
Many patients with autism and other features like some dysmorphic features and other diseases, they always should have this genetic testing performed. And when patient with autism is in genetic outpatient clinic, they had microarrays, karyotype or NGS testing. It depends on the type of autism. It is started often with microarrays and karyotype, and after that, when we still do not know the reason, we perform NGS.
Okay, thank you.
Thank you very much.
That's all. Thank you.
Thank you for your presentation. And now I would like to invite Wojciech Tynior from Medical University of Silesia with a presentation titled Genes Expression Analysis Among Polish Children with Molar Incisor Hypomineralization. Thank you.
Honorable committee, dear friends, colleagues, my name is Wojciech Tynior, and I'm stand before you to present my dissertation project, part of that, Genes Expression Analysis Among Polish Children with Molar Incisor Hypomineralization. In this brief presentation, I would like to share my research findings about gene expression in congenital teeth disease called MIH, or molar incisor hypomineralization. Molar incisor hypomineralization is a general quantitative development disorder that may affect on one or more first permanent molars. It also can affect on permanent incisors. The clinical picture the clinical picture presents circumscribed changes in the translucency ranging from white, green, yellow, and brown discoloration. Affected enamel has a normal thickness. These white spots, as we can see, can occur asymmetrically, and incisors are usually less severe than molars. Here, there's a picture of our patient. You can see discoloration, white and yellowish discolorations on incisors, upper incisors, and the first permanent molar. To give you a bigger picture, I also attached better quality photos. And here you can see opacities, yellow and brownish a bit, and permanent post-eruptive enamel breakdown. That's a huge problem in that case because of masticatory functions, masticatory forces like chewing, the enamel easily can break off. And because of that, it exposes the dentin that is easy to develop caries or decay. The etiology of MIH is highly complex, and scientists said that it's multifactorial and not fully explained. However, we see that general health during first 3 or 4 years can be has a major impact on that disease. In 2022, Garrott and others present systematic review and meta-analysis in which they present three big groups that are responsible for MIH: prenatal, perinatal, and postnatal. They also attach the information that other factors may also occur and play an important role. In the discussion section, they added that and analyzed papers concerning amelogenesis-related genes and add that epigenetics might be the clue. So it can be the connection between environmental and genetical factors. The aim of my study was to analyze presented genes expression in the buccal epithelial cells and teeth tissue of patients with MIH and the control group. Mainly, I was focused on genes that are responsible for amelogenesis, but I also added 2 of genes that are responsible for pain response. And the material and methods present as follows. Firstly, we performed an interview and dental examination to evaluate teeth morphology of our patients. It was done in Uniwersyteckie Centrum Stomatologii I Medycyny Specjalistycznej w Bytomiu. Then we classified our patients to 2 categories: study group, children diagnosed with MIH, and control group, children without MIH. Tissue sampling protocol was the collection of oral mucosa specimens, and if we need to extract the tooth, the teeth were collected and stored in sub-zero condition. Then we performed an RNA isolation using a universal RNA purification kit, and final analysis was performed by real-time PCR using Tachman gene expression assays. And our results are: we obtained 55 children, 27 children with MIH, and 33 children with healthy control. The mean of age and the gender distribution is presented. Interestingly, only 2 of these genes presented activity, expression. However, only 1 of them, TAFT1, presented statistical difference between study and a control group. According to teeth samples, because of ethical consideration, we just obtained 14 teeth, 1 tooth from a patient with MIH, and 6 teeth, first permanent molars with caries, and 7 teeth, third permanent molars without caries. The mean of age and gender distribution is presented. In this tissue, only 4 of these genes presented activity, presented expression. However, only one of them, MMP20, presented statistical difference between control group with carriers, control group with carriers and without carriers. To sum up, I would like to tell that our research is pioneering, and these results provide a new step towards gene expression and analysis among patients with MIH. Statistical significant difference in TAFT1 gene is very interesting finding, but it requires further analysis. And we encourage other scientists to develop that topic and validate our results in a big field on a larger scale. Thank you for your presentation.
Thank you for your presentation. Could you tell, did you receive any funds?
Actually, no. I asked an NCN 3 times and they rejected my project. So I kindly understand that they will not prepare for me any money. Thank you. Thank you.
Okay, and the samples, you've extracted the teeth, the healthy one also?
That's quite interesting topic. Thank you for that question. Can we skip to? Okay. That's very difficult and very important question to analyze. Oh no. Okay. So generally, because MIH is a situation that we have teeth that are really badly destroyed. And in some cases, we have to extract the tooth. So that's our sample. And here I have one patient that was a girl that we extracted 4 teeth because of MIH. So that was very tough for me as a dentist and very difficult for the mother. However, to compare that result to the control group, we cannot extract healthy tooth from a healthy child. We have to obey that limitation, and we generally took third molars, so wisdom teeth. They are quite often we extract in the during our 20s. We extract these teeth, and to have different control, we extract very badly destroyed teeth, so the first permanent molars. But as you can see, the age is really high. So these patients were quite older, and that's our strong limitation of that study, that we cannot compare it precisely. Because of that age. That was a girl, and here adults in middle 20s.
Okay, thank you.
Thank you very much for a really interesting presentation. And I think we can get back to the presentation classification of heart projection in trans-thoracic echocardiography using artificial intelligence methods. Is Gabriela Orzeł with us?
Yes.
Okay, great.
Good morning. Sorry for my delay. Okay, so I have pleasure to present for you the first achievements during my first year of doctoral school. The topic of my doctoral topic is classification of heart projections in transthoracic echocardiography using artificial intelligence methods. Okay, so artificial intelligence integration into cardiology could prove transformative in diagnosing cardiovascular diseases, offering speed and precision surpassing traditional methods. AI methods in TTE are usually used for improvement improvement the quality of images, classification, segmentation, and in advanced cases in diagnostics, because there are models which can give the proposed diagnose, for example, of abnormalities in the morphology of the heart. So, in adherence to the PRISMA guidelines, we conducted a systematic review as a qualitative assessment. Our search strategy for pertinent articles encompasses exploration across two databases, PubMed and Web of Science. The basic search query is included on slide, and we can see that it's very complex. So the selected keywords encompasses the diagnostic methods employed to assess patients like echocardiography, POCUS, with acute symptoms, so patients with acute symptoms like chest pains, dyspnea, and the incorporation of artificial intelligence into this procedure. So yes, and we performed the textual analysis of all results or of original studies, and at the end we had 15 studies included in the qualitative synthesis. In the systematic review, the researchers used several approaches to evaluate cardiac images. First, a classification. A more advanced process was segmentation and identification. So distinguishing important areas into heart. And the last approach was to determine continuous values, like, for example, ejection fraction. The most widely studied parameter was the left ventricular ejection fraction. Because I'm taking a comprehensive approach in my dissertation, the first step is to develop a model to classify cardiac projection, as this is the key component of the study for further image analysis. Okay, so therefore, I will briefly discuss the heart projection classification model which we developed. So convolutional neural networks were used because it is the most suitable method for image processing. The classification model was developed based on training data and validation data. The training data couldn't be the same like the validation data, so these groups must be different. First, I analyzed the given images and assigned them to the name of the heart projections. Oh, I'm sorry. Yes. So first, I had to analyze the images and name the projection of the heart. Next step was to divide video into individual frames. So, for example, this is the first one video, and we have lots of frames from only this one video. Then we resized the images due to need to save computer power. The next step was to separate ROI, so it is area of interest, to isolate redundant data from the images and focus only on the tissue and heart cavities. After the proper data preparation, we designed a CNN model. Convolutional neural networks are specialized networks for image analysis that use convolutional layers here to detect local features such as edges and textures. The process involves incremental processing and image reduction to extract the most relevant information, and finally the network classifies the images using a dense layers. CNN's model automatically learned to recognize the key features, which is effective for computer vision application. And the last slide, I would like to present our results. So the graphs illustrate the result achieved by the model classification, and figure 1 shows the approach from the function side, where the highest loss occurred during the first epochs. So the first 4 to 5 epochs, the loss function was very high, but after the fifth epochs, the loss function was equal to training loss. Okay, and chart 2 shows the accuracy that the model achieves during training and then validation. The accuracy on the validation set was about 98% after just five epochs. So the entire training and validation process was with good accuracy. Okay, and that's my first steps during the first year of PhD, and the next steps after this classification model will be creating a for example, a segmentation model for identifying for example, left ventricular or other chambers. Thank you.
Thank you for your presentation. Could you tell us what was your contribution to the study, and did you estimate the sensitivity, because it was the first step of your project, yes? And did you have this data?
Mm-hmm. Okay, so I participate in all steps during this study, because I achieved lots of images from our Department of Cardiology. Then I labeled the projection, so I had to name all videos with the name of the projection, and next step was to analyzing of the literature and create the main idea of the AI methods using in our future studies. So I analyzed the literature, and in this way, I knew that convolutional neural networks have to be used because it's the most suitable method. And with my partner, in this study, we created the model in Python language. So all the steps will be with my contribution. And the second question.
Sensitivity for this method, but I think that it's a first step, yes, and you will continue the research, yes.
Yes.
Could you tell what is the sensitivity of this model? Yes.
Yes. Okay. So it's 98%. Of accurate, so the sensitivity is very high.
Okay, thank you.
Yes, thank you.
Thank you very much for your wonderful presentation.
Thank you.
So, all the submitted papers has been presented. We congratulate all the authors and participants of our session. We hope that you enjoyed this time and that the exchange of views and observation will contribute to increase our medical knowledge. At this point, I would like to ask the president of the jury, Professor Agata Stanek, for a few words to sum up the session.
Dear speakers, dear attendees, in the name of jury, I would like to thank you, all of you, for invited us for this your conference. It was very interesting session. We heard 13 presentation. The level of the session was very high, and I would like to congratulate you your research and encourage you to publish your research in good journals. And I would like to thank for working in the jury Dr. Baudis and Dr. Pacwa for estimation your results. And I would like to also invite you to attend at the doctoral day, which you see will be held on 24 June here in Katowice. Thank you.
Thank you for these words, Professor. At this point, I would like to express the word of thanks to the members of esteemed jury. We truly appreciate your valuable comments, accurate observation, and work on the presentation assessment. Dear participants, we would like to cordially invite you for the decorating and closing ceremony of the SIMC 2024 conference, which will be held in Reality of Theatre on the 17th of May at 6 o'clock p.m. Ladies and gentlemen, the experimental medicine session comes to the end. On the behalf of the Student Scientific Society, I would like to thank you for all participation in the session and wish you a very nice day. Just after session, we would like to invite you all for lunch from 12 o'clock to 1 p.m. o'clock. Thank you very much.
Excuse me, I have just a question. Hello?
Yes, hello.
May I ask the closing ceremony, we will have a link to, to attend it, or because the link on the website is only for this session. So do you hear me?
Yes, I can hear you, but we are not on the closing ceremony.
I know. I'm not talking about now. I'm talking about tomorrow session at afternoon.
There will be no streaming for the closing ceremony. We will inform you about the results by mail.
Okay. Thank you.
Thank you.
Wow.